Hi everybody, I'm Nauruz. I was working as a principal in a matriculation school for about 28 years. Though I was serving as a principal, I had been teaching biology and zoology to classes 10, 11 and 12. I'm so fond of teaching, it's my passion. So today I'm very happy to present this PPT on this topic, molecular basis of inheritance. So what is molecular basis? That refers to molecules responsible for the inheritance. So the molecules are the DNA or RNA. Molecule here refers to DNA or RNA, which are the nucleic acids responsible for the inheritance, or they form the base for inheritance. Inheritance, again, you must be knowing, it refers to transfer of parental characters to offspring generation after generation. Transfer refers to, it's not transferring all the parental characters, only there are certain characters which are heritable, that is transferred. So this is called inheritance. So in this class, we'll discuss about what is DNA and RNA because they are responsible for this inheritance. So these two molecules, DNA and RNA, they are basically nucleic acids. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. They are the two types of nucleic acid found in all living systems. Out of this, DNA acts as the genetic material in most of the organisms. RNA, though not a major genetic material like that of DNA, but it's a genetic material in some viruses like tobacco mosaic virus or some retroviruses. Otherwise, the function of RNA is different from DNA, that it acts as a messenger, that is, taking the message for the synthesis of protein from the DNA or copying the information for protein synthesis from DNA and taking this message to cytoplasm to a particular organelle called ribosome, which is the site of ribosome, which is the site of protein synthesis. So in this way, RNA acts as a messenger. Apart from it, RNA has some additional roles also. For example, it acts as an adapter. There are different types of RNA. One such RNA is called tRNA, transfer RNA, which acts as an adapter, also called as vehicle, molecular vehicle, that is carrying some anticodons which are required for the synthesis of protein. So this tRNA acting as adapter, picking up anticodons, it's a triplet codon containing the message for the synthesis of protein. So carrying or transferring this triplet codon to mRNA, which has codons which match with the anticodons taken by this adapter or tRNA. And in some cases, some RNA acts as structural molecule. That is, in ribosome, it acts as or it functions as the structural molecule. And in some cases, RNA also acts as a catalyst. So catalyst in some biochemical reactions to speed up the chemical reactions. So such a enzymatic RNA is called ribozyme. So ribozyme, a type of RNA, it acts as a catalytic molecule. So apart from acting as a genetic material in some viruses, RNA acts as a messenger, acts as adapter, structural and catalytic molecule. About DNA, it's the polymer of deoxyribonucleotides. Polymer, from the name we can understand that many monomers are there. Many monomers bound together to form polymers. So polymers of what? Monomers, smaller molecules. So polymers are the macrobiomolecules which are formed by some small molecules called monomers to form these polymers. So nucleotide is an example for monomer. So many nucleotides or more such nucleotides called deoxyribonucleotides bind to form the polymer of DNA. So DNA is called a polymer. Why it is called a polymer? Because it is made up of many nucleotides. So many nucleotides, individually each nucleotide is an example for monomer. So more such monomers or more such nucleotides bind to form this polymer of DNA. So what is a nucleotide? It's a monomer consisting of a sugar, which is a pentose sugar, tricarbon sugar, a nitrogenous base, and a phosphate. So each nucleotide has got three components: pentose sugar, nitrogenous base, and phosphate. So this illustration shows, so this is a single nucleotide. It's a monomer. So more such monomers or nucleotides bind to form DNA or polymer of DNA. So the three components present in each nucleotide is the sugar, the tricarbon sugar called a pentose sugar, and this is a base. Here it is thymine, and this is called phosphate. So these are three components constitute a nucleotide, single nucleotide. Phosphate, pentose sugar, and thymine. We'll discuss in detail about the structure of a polynucleotide. As I showed in the diagram, a nucleotide has three components: a base, nitrogenous base, pentose sugar. Pentose sugar is different in DNA and RNA. In the case of RNA, the pentose sugar is called ribose sugar. In DNA, it is called as deoxyribose sugar. So we'll discuss in the next slide about this ribose sugar and deoxyribose sugar present in DNA and RNA. So these are the three components: base, nitrogenous base, sugar, and a phosphate group. And about the nitrogenous bases, there are two types of bases. One is purine, other one is pyrimidine. Purine base consists of adenine and guanine. Pyrimidine base consists of cytosine and thymine in DNA, but cytosine and uracil in RNA. So in DNA, thymine is present. In RNA, uracil is present. The pairing occurs in such a way that adenine pairs with thymine, guanine pairs with cytosine. This is with reference to DNA, but in RNA, adenine pairs with uracil, guanine pairs with cytosine. So guanine cytosine pairing is same in both RNA and DNA, but adenine thymine is applicable only for DNA. Adenine uracil is with reference to RNA. So this is again the same information about cytosine is common for both DNA and RNA, and thymine is present in DNA. But uracil is present in RNA at the place of thymine. So thymine is present. It is only with reference to DNA. Instead of thymine, uracil is present in RNA. So a nitrogenous base, either cytosine or guanine or thymine or whatever the base, a base linked to hydroxyl group of first carbon of the pentose sugar. So a base any one of the bases pairing with hydroxyl group at the position of the first carbon of the pentose sugar through a N-glycosidic linkage. It is called a nucleoside. So a nucleoside refers to a base and pentose sugar. Base attached to pentose sugar through N-glycosidic linkage. So base is linked to what position? First carbon of the pentose sugar through this linkage called N-glycosidic linkage. So this base and pentose sugar alone, these two form the nucleoside. A base and a sugar, pentose sugar, form nucleoside. So it may be adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. You can understand from the names, adenosine with reference to RNA, deoxyadenosine refers to DNA. Adenosine, that is adenine base of RNA, this is adenine base of DNA. Likewise, guanosine, guanine base in RNA, this is guanine base in DNA, like that. So here you can understand uracil, a base in RNA, thymine, a base in DNA. So this is a nucleoside. So nucleoside refers to a base, any one of the bases with pentose sugar. With this base and pentose sugar, when a phosphate group is linked to hydroxyl group of fifth carbon of a nucleoside. This is also done through a linkage called phosphoester linkage. So with the nucleoside, when a phosphate group is linked through phosphoester linkage, a corresponding nucleotide is formed. So nucleoside refers to a base and pentose sugar only two components. But with this nucleoside, when phosphate group is attached, it is called as nucleotide. So two nucleotides are linked through 3'5' phosphodiester linkage to form a dinucleotide. So when there are two such nucleotides linked together through 3'5' phosphodiester linkage. 3' means which refers to the position of the carbon, third carbon of the pentose sugar, and 5' refers to fifth carbon present in pentose sugar. So two such nucleotides linked together at the 3'5' phosphodiester through phosphodiester linkage to form a dinucleotide. So when there are more such dinucleotides join, it forms a polynucleotide chain. Hope you understood. This is a nucleoside formed by a base with sugar, pentose sugar, nucleoside. So when nucleoside is added with a phosphate group, it is called nucleotide. Phosphate group is linked to hydroxyl group of fifth carbon of a nucleoside through phosphoester linkage. So two nucleotides linked together to form a dinucleotide, and more such dinucleotides or more such nucleotides join in such a manner to form a polynucleotide chain. So this is the illustration to show the two strands of the DNA. So this is one polynucleotide chain. This is another polynucleotide chain. Here it is referred as 5'-phosphate. Here it refers as 3'-hydroxyl. That is, you can understand, with the fifth carbon, this phosphate group is attached. So this end is called as 5'. Whereas 3' refers to a hydroxyl group attached to third carbon of the pentose sugar. So this polynucleotide chain is referred as 5'3' chain, and this is referred as 3'5' polynucleotide chain. Because DNA is formed of two polynucleotide chains which run anti-parallel, parallel of course, but anti, in the reverse direction. One goes in the direction of 5' to 3', other one goes from the direction of 3' to 5'. So here we can understand this is pentose sugar, this is a base called adenine, and this is phosphate. So three components of a nucleotide. So this is a single nucleotide. This is another nucleotide. Sugar, here thymine is the base, pentose sugar and phosphate. So two nucleotides to form dinucleotide. Another nucleotide. So this is called a polynucleotide chain. So here we can understand that adenine of one polynucleotide chain pairs with the thymine of another polynucleotide chain by two hydrogen bonds. So pairing occurs in this way. Adenine pairs with thymine and both are bound together by means of two hydrogen bonds. Whereas between guanine and cytosine, you can understand three hydrogen bonds which hold this guanine and cytosine together. Shall move on to the next slide. So a polymer formed has at one end a free phosphate moiety at 5' end of sugar. Moiety is nothing but it refers to the part of the molecule. So it is not the whole of the molecule, a part of the molecule. So this is called phosphate moiety. So at one end, it is at positioned at 5 carbon of the pentose sugar, 5' end of sugar, which is referred as 5' end of polynucleotide chain. So 5' end of polynucleotide chain means you should understand that is a free phosphate is attached at the 5 or 5th carbon of the pentose sugar. Similarly, the other end of the polymer, sugar has a free OH. That is, pentose sugar has a free hydroxyl group at the 3rd carbon of the pentose sugar. So, hydroxyl group is attached to 3rd carbon of the pentose sugar, which is referred as 3' end of the polynucleotide chain. And in the illustration we had seen, there are two polynucleotide chains. So, in these two polynucleotide chains, the backbone, it is formed due to sugar and phosphate. Sugar and phosphate of the polynucleotide chain acts as a backbone. So the nitrogenous bases, adenine or cytosine or guanine or thymine, these bases are linked to sugar moiety, project from the backbone. So backbone is the sugar and phosphate, and the base linked to sugar, it projects out from the backbone. We shall go back to the previous slide. So phosphate and sugar, phosphate and sugar. So it forms the backbone of the polynucleotide chain. And here also sugar, phosphate form the polynucleotide, backbone of the polynucleotide chain. So, and sugar attached to this base, sugar attached to thymine, sugar attached to guanine, projects from the backbone. So this is the backbone, so backbone of the polynucleotide chain. So we shall move on to the slide, next slide again. So in RNA, every nucleotide residue has an additional hydroxyl group. So in RNA, so this is the main difference between DNA and RNA. In RNA, two hydroxyl groups are present at 2 carbon position in the ribose sugar. So the sugar, pentose sugar present in RNA is referred as ribose sugar. It has additional hydroxyl group at 2' or second carbon of the pentose sugar. One more difference is, major difference between RNA and DNA is, Uracil is found in RNA at the place of thymine. So these are the two major differences between DNA and RNA. Two hydroxyl groups or additional hydroxyl group present at second carbon of the pentose sugar in ribose sugar and uracil at the place of thymine. So these are the two major differences between RNA and DNA. This is the diagrammatic representation to show the ribose sugar. So you can understand here an additional hydroxyl group is seen in the ribose sugar. So this is the fifth carbon, fourth and fifth carbon of the pentose sugar to which a phosphate is attached. So this is the end hydroxyl end with which a base is bound. We'll discuss about this two polynucleotides. This is also an additional slide to show the hydrogen bonds between the nitrogenous bases and sugar and phosphate. So phosphate and sugar form the backbone in both the chains, and in the middle it is guanine and cytosine held together by means of hydrogen bonds. It is 3 hydrogen bonds between guanine and cytosine and 2 hydrogen bonds between adenine and thymine. So this is the difference between RNA and DNA, which already I explained. So in RNA, every nucleotide residue has an additional OH group present at 2' position. Apart from 3' position, another additional group is present at 2' position, second carbon of the pentose sugar. So 2 hydroxyl groups are present at second and third position. One additional one is present at second carbon of the sugar. In DNA, deoxyribose sugar, it has only one hydroxyl group and one hydrogen at the second position, second carbon position, second carbon of the pentose sugar. So here, additional hydroxyl group, but the second carbon of this pentose sugar. Here, only one hydroxyl group at the third position and one hydrogen at the second position. That is why sugar in DNA is called deoxyribose sugar. In RNA, pentose sugar is called ribose sugar because it has an additional hydroxyl group at the second position. DNA is called a deoxyribose sugar because it has only one hydroxyl group. So D refers to one hydroxyl group less, one hydroxyl group lesser than what is present in RNA. We'll move on to the next slide. This is also an illustration to show the difference between deoxyribose and ribose sugar. So in ribose sugar, third carbon and second carbon both that is attached to hydroxyl groups are attached to third carbon of the pentose sugar and second carbon of the pentose sugar. But in deoxyribose sugar, in the second position, hydrogen is attached, and this is like RNA. Third position, one hydroxyl group, but second position is hydrogen. So here, additional hydroxyl group is present. That is why it is called ribose sugar. One hydroxyl group lesser. So it is called, in DNA, the sugar is called deoxyribose sugar. Shall discuss now about the history of DNA. How DNA was discovered. So DNA basically is an acidic substance present in nucleus, first identified by a scientist, Frederick Miescher, in the year 1869. He named it as nuclein. DNA was, when it was discovered, that time it was named as only nuclein, not DNA. Because the scientist must have thought it is present in nucleus, so he named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. That is, the structure of DNA is so complicated, it's such a long polymer, it remains intact. So isolating it was very difficult at that time when technology was not available, not that much advanced. So due to technical limitation, no technology was available. So isolating such a long primer, which remains intact, it was not easy. Elucidation of the structure of DNA, it means not easy to find out or to determine the structure of the DNA. So it remained elusive. It was not easy for the people of scientists for a longer period of time. Then later on in the year 1953, James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. So James Watson and Francis Crick, they proposed a simple but famous double helical model for the structure of DNA, but for that they depended on the data found out by Maurice Wilkins and Rosalind Franklin, two scientists who came out with the data based on the X-ray diffraction of DNA. So it was initially these two scientists, Maurice Wilkins and Rosalind Franklin. So they found out some data based on X-ray diffraction. So depending on this or with this details, James Watson and Francis Crick in the year 1953, they came out successfully with a proposed simple double helical model for the structure of DNA. We know that for the discovery of this double helical or for proposing this double helix model of DNA, they were awarded with Nobel Prize. So one of the hallmarks of their proposition was base pairing. So based on this base pairing between the two strands of polynucleotide chains, it's one of the hallmarks of their proposition. However, this proposition also, base pairing proposition, it was also based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine are constant and equals 1. That is, this is called Erwin Chargaff rule. What he proposed that the ratio between adenine and thymine and guanine and cytosine, it remains constant and equals 1. That is, the amount of adenine and thymine is equal to the amount of guanine and cytosine, and it always remains constant. So this proposition, this pairing proposition was based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine are common and equivalent. We'll move on to the next slide. This is about a unique base pairing property. So the base pairing confers a very unique property to the polynucleotide chains. So this base pairing gives a very unique property to the polynucleotide chains. The way adenine, thymine, cytosine, guanine, the way how they pair with each other, it gives a very unique property to the polynucleotide chains. These two polynucleotide chains are said to be complementary to each other. Complement means mirror of each other. And therefore, if the sequence of bases found in one strand is known, so the sequence of bases present in one strand, if you know that, the sequence on the other strand can be easily predicted. So the sequence of bases in one strand, if it is known, we can easily predict sequence of bases on the other strand. And also each strand from a DNA acts as a template for the synthesis of new strand. So we know that DNA consists of two polynucleotide chains and both the chains are complementary to each other, and each strand acts as a mold or template for the synthesis of a new. So the polynucleotides present in DNA are called parental strands, and new strands formed from each of the polynucleotide chain are called new strands. So the two double-stranded DNA thus produced would be identical to the parental DNA molecule. So the two new strands produced from each of the polynucleotide chain of DNA, they are identical, same like that of their parental DNA molecule. Because of this, the genetic implications of the structure of DNA became very clear. So this is about the base pairing, which gives a very unique property to the polynucleotide chains. So we can understand that the two strands of the DNA are complementary to each other, and each strand can produce its own strand, a new strand. So from the parental strands, new strands are produced. Parental strands act as template or mold for the synthesis of new strands. So the new strands produced are identical to that of parental DNA molecule. Because of this, the genetic implications of the structure of DNA became very clear. So these are the information about what is molecular basis of inheritance. Hope you have understood. And if any doubts, we shall discuss later. Thank you for watching. Bye-bye. Have a great day. Thank you.